Niharika, mute your mic. क्या आवाज आ रही है? Okay. So now I request Poonam madam to start formally this session. Madam.
Yeah, thank you, Gopalji sir, and a warm welcome to everyone. Am I audible?
Yes, yes, you are audible.
Okay. So a warm welcome to all the participants of this Science Dialogue series, and our welcome to our today's guest, Dr. Sumeet Pandey, and I guess this is his fourth or fifth lecture in this Science Dialogue series, as far as I can recall from the whole of the series pattern. Sir is being a valuable guest to us, who has always been with us in departing the knowledge to the students and to the participants whosoever is joining us in this Science Dialogue series. Many of you might be aware about the Dr. Sumit Pandey sir, and few of you who are not very much aware about Dr. Sumit Pandey sir. So just a brief introduction about him. So if I make an introduction, so he is currently an associate director at the Translational Unit Multiomics at GSK in the UK. His research focus basically focuses on the integrating multiomics approaches in genomics, proteomics, and metabolomics to advance the drug discovery and translational medicine. If I talk about his academic journey, so he has earned his PhD from the MS University of Baroda and has various roles at GSK, including scientific investigator, GSK fellow. Before joining GSK, he was a senior postdoctoral scientist in immunology at the University of Oxford, where he explored the role of bacterial induced autophagy in macrophages using advanced techniques like confocal and phosphoproteomics. So, this is just about a brief introduction. And if we talk about his research caliber, so which is very much prominent by the publications of more than 90 articles in various kinds of journal article, book chapters, and so, with more than 1,000 citation. Uh, he has been also awarded global recognition award, silver and bronze category, and also he is an blogger, writes many articles or scientific articles at the Science Circuit blog. So today he will be presenting with us a topic which is of interest to many of us. That is basically targeting fibrosis, advances, obstacles, and the future direction. So just a very brief introduction about him. So his caliber is way beyond whatever I have told in this platform. So Sumit sir, once again, welcome to you.
Thank you,
On the
Sumit ji. Thank you.
Thank you so much for very kind introduction. And I will just share my screen so that we can start with the presentation. Just one sec. I hope you all can see my screen, right?
Yes, it is.
Yes, yes.
Okay, but I cannot see anyone. So
Yes.
if there are any questions, I'm very happy to take
we
in between.
will speak. निहारिका माइक बंद करो या आपको रिमूव करना पड़ेगा निहारिका शर्मा. Yes, please continue.
Okay, sir. So thanks again for the kind introduction. And today I thought I will give you an overview of a condition known as fibrosis, and I will try to cover in next 45 minutes why it is important and what are the knowledge gaps and therapeutic gaps in terms of targeting fibrosis, what technological advances we can use in today's world to understand fibrosis in different disease setting, and what are the opportunities in terms of drug development. But before I go into the details of fibrosis, it is extremely important to understand what is wound healing. So whenever we have any injury, which you can see here, if you have any cut or wound, the first thing what happens is there is a clot formation. And after the clot formation, you do have a lot of immune cells which go to the site of inflammation. The very first cells which actually reach to the site of inflammation are the neutrophils, and then this is followed by macrophages, T cells, and other immune cells. But the whole point of this migration of immune cells is to actually resolve what has happened at the site. And, and this involves a lot of resolution, involves macrophages because they are one of the key immune cell types. So angiogenesis is super important, which is formation of new blood vessels. And then what you can see here is there are some cells which actually proliferate over a period of time. And then there is remodeling and resolution, meaning you can see this structure of the epidermis is now intact. And there are some resolution factor, ECM proteins, and matrix cross-linking protein, which play a very important role. This whole process can take from hours to months, depending on how big the wound is. So within hours, you can have the blood clot, inflammation within days, angiogenesis, cell migration and proliferation within weeks, and then remodeling and resolution within a month. Now, what you can appreciate from this image is that it's a very complex process overall, which involves multiple different cell types, not only immune cells, but also stromal cells, to, to resolve to resolve and remodel where the wound was there. Now, what happens is, if you have the inflammation which is persisting for longer period of time, meaning it's a chronic inflammation, you do have this process which is kind of amplifying all the time. And this is what is fibrosis. So fibrosis is excessive accumulation of connective tissues in an organ following injury or chronic irritation. And this is basically characterized by buildup of extracellular matrix protein, and it also disrupts the normal tissue architecture, and often it leads to organ dysfunction or failure. It has a systemic impact because fibrosis can actually affect every major organ, including lung, heart, blood vessels, liver, kidney, gastrointestinal tract. And within these organs, multiple different indications have the fibroblast or fibrosis playing a major role. There are multiple chronic diseases which involve fibrosis. For example, asthma, COPD, atherosclerosis, IBD, IPF, and many more. Up to 45% of the death in the developed world are actually linked to the fibrotic tissue response, and global incidence and healthcare burden of fibrosis is steadily increasing. And if you would like to visualize this across different organs, what you can see here in this figure is you have a liver tissue and you have a normal architecture of the liver. But when you have non-alcoholic fatty liver disease, or you have viral hepatitis or alcoholic damage, what you see here is a lot of deposition of extracellular matrix. So these are a lot of fibroblast, myofibroblast, which are accumulating within the tissue. If you talk about kidney, this is how the normal structure looks like. But during the fibrosis, which could result from diabetes, proteinuria, or hypertension, or any glomerular disease or crystal deposition, you have a lot of ECM deposition, which you can see here. Similarly, in heart, you have the similar deposition, and also in the lung. And what you can see from here is there are a range of these disease within every single organ, which actually is resulting into fibrosis. So there must be something which is very fascinating about fibrosis. Number one, that it is affecting different organ, which of course means there should be resolution. And it is affecting different organ in a different way, which means organ-specific response play a very important role. So let's go into a little bit more detail.
हाइड कर दे, नीचे जो है उसको हाइड कर दे लाइन को.
What you can see here is the whole fibrosis response and stages. So the very first thing which which happens is inflammation. So this could be as a result of any wound or as a result of any bacterial infection, or it could be a disease specific triggers or cellular site of injury. It could be anything which actually results in inflammation. The second step is the fibrotic tissue remodeling, which basically means that there are different types of cells, and these different types of cells can actually form myofibroblast, and they are the key cells which will be secreting a lot of extracellular matrix, ultimately resulting into much more stiffer tissue. And the third stage is self-amplification loop. Then there is an self-amplification loop, meaning you have cells and somehow they cannot sense that now the inflammation is resolved. So they keep firing up, and there is a lot more extracellular matrix deposition. And over a period of time, your tissue becomes much more stiff. But the fascinating thing here is that you do have some of the distinct mechanism of fibrosis. And there are differences at these three different stages. So for example, in the inflammatory stage, you have disease specific trigger or cellular site of injury, meaning this could be an epithelial cells, or it can be stellate cells, or it could be some of the other cell types, including keratinocyte if it is skin. And the composition of infiltrate, meaning what are the types of cells which are actually going to the site of injury. Similarly, the source of myofibroblast and their relative contribution to fibrosis can be very different. There are different cells which could be differentiated into myofibroblasts. It can be either resident fibroblast, epithelial cells, fibrocytes, pericytes, or endothelial cells. And they all can actually form myofibroblasts, which you can see here with different colors. They come from different sources. And then you have a self-activation loop, which means that you can have some genes which are ECM related genes, and they are very specific to some organ, and you can have some of the other related genes which are different. Now, if you look at the shared mechanism of fibrosis, then again we can categorize these into three different categories. Number one, involvement of immune cells. This is something which is common across all the organs. You can have leukocyte infiltration, release of Th2 or M2 cytokines, or Th2 M2 polarization, and there are these cells which you will find in every single tissue, independent of which tissue fibrosis we are talking about. Similarly, if we look at the core pathways of fibrosis like TGF-beta, Wnt signaling, PDGF pathway, or nuclear receptor signaling, granulocyte cyclase, or 5-HT signaling, this is something which is again shared across different organs. Tissue stiffness, hypoxia, and epigenetic modification is again something which is shared across diseases. And there are some evidences from clinical practice. So for example, if you consider inflammatory bowel disease, there are cases of stricturing Crohn's disease, meaning this is the disease which is having more of a fibrosis component. And the only solution in today's time is basically operating the fibrotic tissue. But once it is operated, the fascinating thing is it will again come back after a few years, which means that there are some epigenetic modifications which are still there in the adjacent tissue, which results in the formation of fibrosis over a period of time again. Although this is the case of intestine, so of course you can chop it and you can undergo surgery. It is much more easier, and this is the go-to method, or you can say a standard of care currently. But if it is happening in the other tissues, for example, like in lung or in kidney or in liver, these become much more challenging because you don't have option of just chopping it off the piece of fibrotic tissue. So if we specifically now focus on the lung and just try to understand what exactly is happening in the lungs, so we have normal lung, which you can see here. These are the cells with very few fibroblasts, which are which are seen here. And the lung capacity in terms of respiration and functionality of lung is pretty normal. When there is injured alveolar epithelial cells, this actually results in activation of immune system. So you have alveolar macrophages, neutrophils, eosinophils, which actually are activated because of the injury. And they release a lot of cytokines which promote the myofibroblast formation. So TGF beta, which you can see here, some inflammatory cytokines like TNF alpha, IL-1 beta, and they can affect the lung fibroblasts. So these are the resident fibroblasts, which you could see here. And these lung fibroblasts, they differentiate into myofibroblasts, and these myofibroblasts secrete a lot of fibronectin and collagen. And due to this, what is happening is your lung tissue is getting much more stiff. The capacity of lung to exchange the oxygen CO2, it drops, and actually it is life-threatening. Similarly, all these cytokines can also affect epithelial mesenchymal transition. So you can see these are epithelial cells, but they can be converted into the mesenchymal cells. These are fibroblasts, and they can also be converted into the myofibroblast population. So ultimately, myofibroblasts are the culprits. Okay, so once the myofibroblasts are formed, meaning that they will secrete a lot of fibronectin, collagen, and it will result in ECM deposition. But there are few things which are really interesting in this, and I will come to that in a minute. But before that, I just want to highlight that this field is extremely complex. Okay, what you can see here is different pathways which have been targeted therapeutically over several years, over decades. Okay, and we have only 2 drugs which have been approved for IPF or for fibrosis, which are here, pirfenidone and nintedanib. And this is both for IPF. And the interesting thing is, even after taking this drug, the survival of patient is only for 2 years. Meaning, if someone is having IPF, and if you give the best standard of care which is available right now, the survival of that patient will last only for 2 years, which means we need to do a lot of work in order first to understand what is the cellular process or biological pathways which are driving fibrosis in different conditions. And second, what are the therapeutic target which could be potentially developed in order to fight any fibrosis across indication. People have tried different anti-inflammatory agents like corticosteroids or JAK inhibitors or IL-targeted therapies. People have also used some anti-fibrotic agents. These are mainly in the pipeline, for example, galectin-3 inhibitors or anti-TGF-beta antibodies or integrin antibodies. And people are also thinking of targeting both inflammation and ECM remodeling because, as I showed you in the previous slide, that there are 2 components. Number 1, you have chronic inflammation which persists over a period of time. And number 2, you have the ECM deposition which results in a more of a fibrosis tissue. So if we can block the pathway which is resulting in ECM deposition, at the same time, if you can restrict the chronic inflammation, then that sounds like a very promising approach. But so far, we have not been successful in doing it with efficiency. So the question is, what is the key therapeutic gap? As I mentioned in the previous slide, pirfenidone and nintedanib, it actually slows the disease progression, but the limitation is they have modest efficacy. They lack the therapy targeting fibrosis regression mechanism, and there is a lot of heterogeneity among patients, which complicates the whole treatment. Which also means, because we do not have any drugs which can actually regress the fibrotic mechanism, meaning if you have fibrosis, it could not be reverted back, which suggests that we need to target fibrosis at a very early time point rather than waiting late. In terms of challenges, there are diversity of cell types and signaling cascade, which make it challenging to understand the key mechanism and effective therapies targeting fibrosis, which you can see here. So for example, if we talk about the hepatic fibrosis, It can be initiated because of hepatitis virus infection, or it could be because of alcohol, or it could be because of the metabolic disorder. And you have these hepatic stellate cells which are getting activated, and they actually differentiate further into the myofibroblast population. If we talk about the pulmonary fibrosis, we do have fibroblasts getting converted to myofibroblasts, but at the same time, you have macrophages, which is resulting into activation of the other immune cells and ECM deposition. You have some epithelial cells getting converted to the myofibroblast population. There is a huge component of cellular senescence, meaning that you have a natural aging process where your cells, over a period of time, they actually have a reduced capacity to function optimally. And one of the key hypotheses is also that when you have any disease or any chronic disease, you are actually aging much more faster. So if one can target the senescent cells, then it gives you an opportunity on the top of targeting some fibrotic mechanism, you also clear some senescent cells because they are the key source of chronic inflammation. At the same time, what people have shown is that many drugs which show some antifibrotic efficacy in the clinical trial, but the mechanism underlying their ability to prevent the injury to the normal cell is still unknown. So we don't want to target anything which could actually affect the normal cell and the normal physiology of any organ. So these are some of the bigger challenges which the whole, you can say, the academic world and the pharma world is currently facing. But there are also some opportunities where we can use some cool technologies to answer some of these questions. Now, in terms of opportunity, there is always a hope because it's a very complex disease, but complexity comes with opportunity. So I have put here a figure which you have seen in the previous slide, talking about different stages of fibrosis. And what you can see is if we can identify what are the key immune cells which are initiating the fibrosis or initiating the fibrosis cascade, then we can target some immune cell types. If we know there are specific fibrocytes or pericytes, endothelial cells or epithelial cells which are resulting into myofibroblast formation, then one can think of targeting specific fibroblast population. And we can also use this as a fibrosis biomarker, because if we know from the cell type markers what are the cells which are resulting into the myofibroblast formation, we can target specific cells without touching some of the normal cells. The third thing one can do is the fibrosis biomarker, which is at the end stage. So what is really important in the drug discovery is not only the development of drug, but also to have a proxy readout which can tell you about the disease progression. And if you are giving a particular drug, that drug is working. So you need to have some sort of readout to say that your drug is working or it is not working. And one of the way you can do this is looking into tissue stiffness, for example. So if after the treatment, the tissue stiffness is reduced, that is a good indicator that your drug might be working. And depending on the organs, you can have different challenges. So for example, if you have skin fibrosis, then it is much more easier to take the skin sample. But if someone has a lung fibrosis, it becomes much more difficult because the bronchoscopy is a very painful process, and everyone won't give you the biopsy sample all the time to actually look into the biomarkers. So there are some proxy ways to look at it. There are some Fab-based imaging, which is basically looking at some of the fibroblast activating protein and then doing the imaging and looking at whether the fibrosis progression or the fibrotic tissue is reducing over a period of time. But we do have to investigate this. There are some single cell RNA-seq enabled high resolution profiling of fibroblast population, which is really good because currently we do not understand fully from where these myofibroblasts are coming from. We know that there are myofibroblasts, but because we don't have great cell-specific markers, we cannot trace it back from which cell types they are originating from. So single cell study can actually enable us with the high resolution profiling of fibroblast population, and it can offer a detailed cellular atlas of how fibroblasts contribute to the fibrosis. But most of the studies till date, they are focused on CAFs, which are cancer associated fibroblasts. And you can imagine it's very obvious why people have focused on cancer-associated fibroblast, because if someone has cancer, most of those tissues are removed, so tissue are much more easily accessible, and you can actually apply all these high-end technologies. But for many of the chronic diseases, tissue availability is a challenge. So just highlighting three challenges. If we have all this, then what is missing and why we are not progressing with the speed we would like to progress? Because there is lack of longitudinal data set to track changes over a period of time. So if I go back to this figure here, I mentioned that when you have inflammation till the tissue resolution, you have a window of hours to months. Okay. Now, if you apply any of the technology, most likely you are looking at a snapshot at a given time point, meaning you might be looking at most likely at the later stage of the disease. And if you're looking at the later stage of the disease, most likely you are looking at the ECM amplification stage, or you're looking at the fibrotic tissue remodeling stage, but not more into the immune cell involvement, because immune cells might come at the very start of the process, but then they disappear. But if you have a snapshot, that actually limits your capacity to investigate the role of those cells. So longitudinal data set is a major data gap in multiple different indications. Now, absence of internal control for robust comparison is another missing piece, which means that what we call as a normal tissue. What is the definition of a normal tissue? And often when people are taking these tissues, they are taking it from the inflamed side or the disease side or the cancer side, but they are missing the adjacent tissue, which might be a non-tumorous or non-inflammatory or non-inflamed tissue, which is just a tissue which is next to the inflamed side. Now, this is extremely important, because if we are collecting the samples from patients who are healthy versus the disease, yes, you have a good control, but because the human population is so diverse, you need a very high number of control samples and also disease samples to make a robust comparison. But if from the same patient, you could collect the inflamed tissue and also the tissue which is adjacent to the inflamed tissue, it provides you with a robust internal control which one can use to understand the disease biology. Apart from this, the other key thing which is missing is the limited insight into the drug response. I'm sure you might have noticed from last 3 to 4 years, there has been hundreds and thousands of papers who are talking about the cell atlas of this, cell atlas of that organ, cell atlas of this particular disease, cell atlas of something else. Now, this is great because what it offers is that we have a better understanding of the cell types which are there. We have better understanding of the developmental processes. We have better understanding of how the control tissue looks like in comparison to the diseased tissue. But what is really important is that we also understand that if someone has been given a particular drug, which, say, for example, reduces fibrosis, can we have access to those tissues? Because that will help us to understand, if you give a particular drug, what are the effector cell types and how those effector cell types are actually causing the resolution of the disease or decreasing fibrosis. This is something which is missing, and there are collective efforts which people are putting together to fill these three gaps. So what I want to do now is, because I have given you some idea about what fibrosis is and what are the bigger challenges, what are the missing links in order to connect this, I just want to highlight one paper, which is on spatial transcriptomic characterization of pathological niche within IPF, which is idiopathic pulmonary fibrosis. And what you can see here is that in this study, they have taken the control lung tissue and they have taken the IPF lung tissue, which you can see very clearly here from the histology image. And then they actually take this tissue and put it on the slide to do the spatial transcriptomic. And I will explain you in a little bit more detail what exactly that means. But for now, uh, just take my words that they have taken the tissue sample and they do something with the tissue sample so that you have this colored images, which you can see here. And every single dot which you see is a single cell. And the key point is that here you have a piece of tissue. So you're not dissociating the piece of tissue. You literally have the whole piece of tissue, meaning you have blue cells here, which are one specific type of cells. You have green cells, which are a specific type of cells. You have orange cells, which are different types of cells, and so on. So they have used 11 samples, and they have used 57,787 spots, and every single spot is a single cell. And they have detected almost 12,500 genes in total. There are different platforms who can do the spatial transcriptomic, and one of them is from 10x Genomics, which is the 10x Xenium platform. And this 10x Xenium platform provides you a very high resolution image of genes expressed within the tissue without even dissociating the tissue. So literally you can have it like a 2D map, which you can see here, and you can look into what are the genes which are being expressed literally at every single spot. And you can complement this technology by something known as COMET, which is sequential high-plex protein immunofluorescence. And in this, you can actually have antibodies which are targeted against specific protein, and you can see the gene expression at, both at the mRNA level and at the protein level. What this study also does is they use some of the 6 publicly available single-cell RNA sequencing dataset, and they integrate and annotate all of them to have a good understanding of what are the different types of cells. And the single-cell sequencing is more of an untargeted type of an approach, meaning that you are not pre-selecting the genes which you are interested in. Xenium platform is more of a targeted platform, meaning you can look at the gene expression from 500 genes to almost 5,100 genes, but this is not an unbiased approach of looking into the gene expression. And once you have all this data set, it actually needs a huge amount of computational power to visualize the every single slide. So there are some tools which are Visium slide integration, meaning that they actually look at or analyze every single spot, which you can see here, and they tell you about which genes are being expressed. They also tell you about the cell types, the pathology. They also tell you about spatially how these different cells are interacting and so on. I won't go into details of that because that's much more complex. But for now, just remember that we have control tissue from the IPF patient, we have the IPF patient lung tissue, and we have done something which is called as spatial transcriptomics. So for those of you who are unfamiliar with the single cell technology, I just want to give a quick introduction. So single cell omics, as the name suggests, it's the technology which allows researchers to look into the individual cells. Okay, so you can literally look into the individual cell gene expression. If I give you an example, you can have a tissue, and this tissue is a piece of tissue which is composed of multiple different cell types, which you can see here. You have a red cell, you have blue cell, you have green cells, and these are isolated, and these are isolated cell types. These are sequenced, and once you have the sequence, you literally have every single cell and the genes within that single cell. What you get as an output is like an Excel sheet. So you have cell 1 and all the genes which are expressed in that one cell. Then you have cell 2, then literally all the genes which are expressed in the cell number 2, or cell 3 and cell 4 and cell 5 and so on. And what you can get from this is you have the genes on the y-axis, you have cells on the x-axis, and then you can apply some principal component analysis, and you can cluster the cells which are very similar. Now, these cells are clustered together because computationally, you can cluster those cells based on the genes they express. So for example, there will be some B cells which are expressing B cell specific genes. There can be some macrophages or epithelial cells or T cells, every single cell types, they have some genes which are very specific to those cell types. So the way you can do this programming and computational analysis, you can actually pinpoint what are those different cell types, and you can segregate single cells. And this actually gives you insights into cellular heterogeneity, which bulk method will not give. So the bulk RNA sequencing, which most of you will be aware of, is the technology where you just take the piece of tissue, you homogenize everything, and because you homogenize everything, you have mRNA from all the cells which are present in the tissue, and you will not get the information at a single cell level. So this is the key advantage of this technology, that you can actually look at the individual cells. Now, the second technology which I mentioned is the spatial omics, meaning this is the technology which keeps intact the spatial relationship. In this, we are not doing any tissue dissociation. So if you just look into this figure here, you have a brain tissue. You take a piece of tissue, you do the sectioning with a microtome, and you put it on the slide. Slide has oligo-DTs and some spatial barcodes, and you can see there are different spots, and every single spot have these oligo-DTs and spatial barcodes. And this actually does the mRNA capture. So there is a lateral movement of mRNA, and you can capture them on the slide. And what you get is you get a similar type of spot cells, meaning every single spot here is a single cell. And then you can look into the gene expression of every single spot without dissociating the tissue, which means this technology, in addition to the previous technology which I mentioned, keeps the cell location within the tissue. Meaning if I have dissociated the tissue, I can get the cells, but I don't know where exactly these cells are located within the tissue. But with this technology, you have a slice, you're not dissociating the tissue, and you can keep the spatial information intact. What it allows you to do is, it helps you to do the image analysis. You can identify your cell types, you can deconvolute, you can identify spatial distribution, meaning you can see this is the area which is purple, this will be one type of cells. You have green, this is another type of cells, orange, this is another type of cells. And what you can also do is you can look into the cell-cell interaction, that how your macrophages are interacting with fibroblast, or how B cells are interacting with fibroblast, or macrophages interacting with T cells, and so on. The most important thing is you can also look at the subcellular localization. So within a single cell, you can also do some subcellular staining with some antibodies, and you can identify within a single cell where exactly your gene is being expressed. And the most cool part of this technology is that you can have the histology image, which is a normal H&E staining on any tissue, and you can take the H&E staining and you can overlap with the transcript. So you have on a slide, you have the information of the mRNA, and on the H&E staining, you have just simple H&E staining, and you can overlap this. And what it allows you to do is, with histopathological staining or H&E staining, you can identify the areas which are fibrosis areas, or you can identify the areas which are inflamed areas. Now with the transcriptomic, you can see this is a very identical image. Now you can look into this part of the tissue, and you can identify what are the genes which are expressed in specifically in this particular region of the tissue. This is extremely important because you have to think from a point of view that if there is an inflammation or tissue damage or anything which actually results in the compromised ability of your organs or cells to function, you do have some cells which are behaving odd. It's not the whole organ. Now, when you do the analysis at a bulk level, or you consider the whole tissue, you might not be seeing the changes which are happening in a very spatial niche location. And this is what this technology enables. Because you have it on the slide, you can have histology slide, transcriptomic slide, and you can identify within a pathological niche or within a fibrotic niche what are the cell types and what genes they are expressing. So this actually brings me to the paper which I mentioned before. that there is a control and the IPF lung tissue, and they have run the spatial technology. They have done Visium and they have done Xenium. The only difference between Visium and Xenium is untargeted, Xenium is a targeted technology. So meaning you can detect somewhere between 500 to 5,100 genes. And the COMET technology, which is basically looking at the spatial proteomics with the help of antibody. Once you have these results, now you can appreciate how powerful it is. On the top row, you have the control tissue, and on the bottom row is the IPF tissue. If you look at the 81 cell abundance in the control tissue, you can see everything is almost blue in color, suggesting that one of the key cell types which are super abundant in the lung tissue is actually 81 cells. When people have IPF, which is idiopathic pulmonary fibrosis, you can see these cells are almost depleted from the lung. Not only this, we know that myofibroblast is one of the culprit cells which results in fibrosis. And what you can see from this image is there is almost no myofibroblast in the control condition or in the healthy tissue. If you look at the IPF tissue, you have lots and lots of myofibroblast. They are side of the tissue. And one of the cool thing is that IPF actually progresses from the periphery to the internal side. So this actually confirms that you have these myofibroblast formation, which is actually at the edge of the tissue. And then you have a lot of myofibroblast, which are abundant in IPF, but not in control. Similarly, there are some specific macrophages, which are known as good macrophages. They are FAB P4 positive macrophages. They are having heterogeneous distribution across the tissue. And when the patients have IPF, you actually see much more decrease in these FAB P4 positive macrophages in the IPF patients. If we go into a little bit more detail and then we try to quantify, because I mentioned that every single spot is actually a single cell. So technically speaking, you can now count the spot which belong to a single cell or a one particular type of cell. So what you can see in this image here is you have a control tissue and you have an IPF tissue. And these colors are suggesting different types of cells. So there are airway cells, there are alveolar cells, fibroblast cells, fibrotic cells, immune cells, alveolar macrophages, and other macrophages, smooth muscle cells, and so on, and so on. What you can appreciate here is in the control, you have lots and lots of alveolar cells, which are these cells, 81 cell types, and they are actually decreased once you have fibrosis. Also, the most interesting thing is, if you look at the red color here, which corresponds to the fibrotic tissue, you actually don't have fibrotic tissue in control, but once you have fibrosis, you can actually have a lot of fibrotic tissue, and you also have component of fibroblast, which is increasing in the tissue, suggesting that there is more of a tissue stiffness and more of an ECM deposition, and most likely they will be converted into the myofibroblast. What you can appreciate from this diagram here is this is again the control tissue. And now, because we can label every single spot with a specific cell type, you can see how and where your fibrotic cells or immune cells or airway epithelial cells, where exactly they are located. One of the very clear difference, what you can observe from this panel versus this panel is, in the bottom panel, you can see a lot of red color across the tissue. These are all fibrotic tissues. These red things, they are all fibrotic. What you can also appreciate is there is a lot of green color and smooth muscle cells which are abundant in the IPF tissue, and this gives you a little bit more insights into where exactly your fibrotic tissue are and in which proximity they are sitting. Are they interacting more with the fibroblast, or are they interacting more with the T cells or macrophages or B cells and so on? So this analysis could be done at a very granular level, and you can do depending on the question. But one thing which I just want to highlight is SPP1 positive macrophages. So they are localized within the fibrotic airway lumen. This is the image which is showing you the spatial distribution. And this is just showing you the different colors. So for example, you have macrophages, which are CHI3L1 positive and CD9 high macrophages. And these are the macrophages which in this study they have labeled as actually SPP1 type of macrophages. And what you can see is you have these niche, which is a very specific niche, which is a fibrotic niche. And you can try to validate this not only at the transcriptomic level, but you can also try to do this at a proteomic level, where you can have these SPP1 positive macrophages, which are mostly CD68 positive macrophages, and they are actually localized within the fibrotic niche. So in this study, it actually gives first time the spatial context and where are these fibrotic niches within the tissue, and what are the key cell types which are actually interacting, and what are the cytokines or receptor ligands which are being expressed by these immune cell population. Once we have this, actually there are multiple different ways of targeting. And here I have focused more on the fibroblast, but there are other methods and ways to target some immune cell population. But I thought, because the fibroblasts are one of the core cells which are involved in multiple different indication, people are actively thinking of actually depleting the fibroblast population as one of the key strategies to develop drugs and to resolve the disease. And what you can see here is, if you have a fibroblast population which you know about is very specific to a particular disease and is increased in an inflammatory condition, one can use some of the antibodies which are specifically targeting some of the cell membrane protein expressed by the fibroblast, or it could be done by using some of the monoclonal antibodies with the CAR T cell type of approach or cytotoxic T cell type of an approach. One can also target some intracellular signaling like Notch3 pathway, and there are some monoclonal antibodies which people are testing to target a specific fibroblast population in a preclinical model. One can also target inhibiting proliferation and migration. So fibroblast, recent publication from Chris Buckley's lab based in Kennedy at University of Oxford suggests that there are some migratory type of fibroblast and there are some resident type of fibroblast. And one of the key thought process is that if you actually inhibit the proliferation of these fibroblasts, then you can reduce the stiffness of the tissue. I mentioned you about the senescence. That's something very interesting. People are also thinking about inducing apoptosis in these cells and modulating metabolism in fibroblast or inhibiting some intracellular signaling. So there are a range of things which needs to be, which can be potentially used as a targeting strategy depending on what type of cells we are interested in. Are we confident about a specific cell type marker which we are interested in, or are there any organ-specific cells which are the real culprit cells in the whole process of fibrosis? Now, having said that, I just want to say at the end that you do have opportunities. There are some emerging technologies which is giving more insights into not only what are the different cell types, but also having a spatial resolution of the tissue provides an opportunity to investigate that what are the key cell-to-cell communication mechanisms which could potentially be targeted to develop drugs which can target fibrosis across indication. So just to summarize, fibrosis is a complex interplay of chronic inflammation and tissue remodeling. There are advances in molecular understanding, and there are some studies basically developing some novel therapeutic strategies. Mostly this is in a preclinical space, and actually a couple of them are also advancing to the first time in human. There are key challenges. For example, early diagnosis is one of the biggest issue, because most of the time we do have late diagnosis. There are targeted therapies, which means that we would like to target one specific population or individualized care. This is a future, but is still far away, because these technologies and targeted therapies are very expensive, and they need to be personalized, and that's not affordable to the major population around the globe. Future lies in multi-targets. So for example, patient-specific approach integrating the biology and technology. And I hope I have provided you with a bit of flavor of how biology and technology could be combined together to get the disease insights. And those disease insights are extremely crucial for developing new therapies for the patient. So thank you so much for listening, and very happy to take any questions.